My name is Lucas Myers, and this is Personalized Medicine, the Future of Oncology. So, what is personalized medicine with respect to cancer? Personalized medicine is a new approach being used towards cancer treatment that uses a patient's genomic profile to guide decisions made regarding prevention, diagnosis, and treatment of the disease. So, a new biopsy approach being used is cfDNA liquid biopsy. So cell-free DNA or cfDNA liquid biopsy is a new way of testing for the presence of cancer cells using modern genomic technologies. Liquid biopsy uses a bodily fluid sample that is non-invasive to collect. So a cfDNA sample is analyzed for the presence of cancer cell biomarkers. So what's the procedure? So first a sample is taken. Typically this sample is blood. And then within the sample, we'll have cfDNA as well as proteins, tumor cells, and probably like RNA. Then we'll amplify certain biomarkers and also like oncogenes. And we'll do this using PCR with the specific primers. And then we'll analyze the results. So from the analysis, we'll be able to determine if cancer cells are still present within the body. And we'll also know which specific biomarkers are present, but more importantly, which specific oncogenes are present. So why would we want to do this liquid biopsy? So tumor cells are subject to evolutionary forces, which can make the genomic profiles of the tumor hard to determine, and also will change over time, and they can also differentiate from each other. So this will result in a heterogeneous collection of cancer cells in terms of a genomic profile. A single lesion biopsy approach is not suitable. It's invasive and it does not collect enough information, especially if metastasis has occurred. So also like keep in mind that as these cancer cells may change over time and change among different places if metastasis has occurred, surgery will no longer be useful because it won't be able to collect all the different kinds of cancer cells. But also over time, we can't do surgery more than once, especially in multiple parts of the body. So that's why we'll need a new biopsy approach like liquid biopsy. So cell-free DNA analysis by liquid biopsy shows promise for identifying simple biomarkers and multiple clinically relevant resistance mechanisms among the tumor heterogeneity. So liquid samples are then processed using PCR for cancer-specific cfDNA quantity, as well as the presence of oncogenes. Liquid biopsy can be used before diagnosis and during diagnosis and following treatment. So you'd want to do it following treatment just to make sure that you've got rid of cancer cells. And you can do it during diagnosis to check for like the evolution of cancer cells. And then maybe we'll need to switch up which therapies we use. So now we'll go into some specific biomarkers and oncogenes. So we'll start with PIK3CA mutations. So this gene is a part of the rapamycin pathway used in intracellular signaling and cell proliferation. So mutations cause a gain of protein enzymatic function and induce oncogenic transformation when expressed in chicken embryos. So in a phase 3 trial enrolling PIK3CA mutant luminal breast cancer patients, adding the specific drug alpelisib, which is a P110 alpha inhibitor. So P110 alpha is a subunit of the PIK3CA protein, to an estrogen receptor antagonist, which is already used as a cancer drug. We've seen significant improvement in anti-tumor activity. And then this gene is like found in one specific spot in the human genome, so the location is noted. So what are the uses of this mutation identification? So identification of the PIK3CA mutation can be used as a biomarker for the presence of cancer cells. But the PIK3CA inhibition is also showing signs of a therapeutic target. So next we'll move on to BRAF mutations. So the BRAF gene plays a role in cell growth by sending signals inside the cell, promoting cell division. BRAF mutations are major oncogenetic drivers in 7% of solid tumors. So the BRAF V600E, referring to the 600th amino acid being converted from a valine to a glutamic acid, is the most common BRAF mutation. And there's a treatment for this specific BRAF mutation. So in a phase 3 trial, 665 metastatic colorectal patients with the BRAF V600E mutation received 3 drugs. I won't say their names because I'll butcher the names, but compared to a chemotherapy treatment, this new treatment showed a better response rate than chemotherapy, so 26% versus 2% response, as well as a significant improved overall survival. So once again, this gene is found in a specific location, so that location is noted on the slide. Anyway, this new treatment, because of its success, allowed for the treatment to become a new standard of care for this specific mutation when present. So what are the uses of this mutation and identifying the mutation? So it can be used as a biomarker. So simply identifying the BRAF mutation can be used as a biomarker for the presence of cancer cells in the body. But then it can also be used as a target of interest. So BRAF inhibition is showing a sign of being used as a therapeutic target. So another one, neurotrophic tyrosine receptor kinase fusions, so NTRK fusions. So fusions involving NRTK, along with a wide variety of other genes that act as fusion partners, have been identified as oncogenetic genes. So the neurotrophic tyrosine receptor kinases make up 3 receptors with a tyrosine kinase intracellular domain able to activate signaling pathways, which then results in things like enhanced cell proliferation, which is a cancerous feature. So the fusion is when the 3' region is fused with the 5' region of the fusion partner, so resulting in a chimeric receptor protein that will be forever activated, resulting in an oncogenetic gene. So things like like uncontrolled proliferation. And then because it's a fusion, the location of this oncogene is could be different every time. It has the potential to be different, especially since the fusion are with like multiple other genes, so the location is not noted, but we can still detect it with like CF like DNA liquid biopsy. All right, so then NTRK fusions have been detected as possible therapeutic targets. So adults and children who possess the NTRK 123 fusion were treated with leratrestinib, which is a selective TRK inhibitor. The overall response rate was 75%. So now, what are the uses of NRTK fusion, like identification? So once again, it can be used as a biomarker. So identification of the fusion can be used as a biomarker for the presence of cancer cells. But then once again, the NRTK inhibition is showing signs of being a therapeutic target. So moving on to another biomarker and oncogene. So fibroblast growth factor receptor pathways, or FGFR. So all FGFR receptors participate in cell survival and proliferation. So deregulation of these receptors is implicated in oncogenesis, tumor progression, and resistance to anti-cancer therapy. So FGFR alterations range quite a lot. So there's really like there's no specific alteration that's like more common than the other. That's kind of the problem with this current marker. But there are a few therapies. So an FGFR4 tyrosine inhibitor named erdafitinib demonstrated anti-tumor activity, achieving an objective response of 40% in previously treated patients. So patients that had already gone cancer therapy, but which was not completely successful, got a 40% response rate from this new treatment. So FGFR alteration uses. Once again, FGFR alterations can be used as biomarkers for the presence of cancer cells. But once again, they can also be used as a therapeutic target. Now, they also, because these alterations range so much, I'll emphasize target of interest a little bit less because, for example, like the BRAF V600E is like a super common mutation, whereas all these alterations are a little bit less common. All right, so further thoughts and for the future. So all the previously mentioned biomarkers were oncogenes. So when their presence is detected, we now have like specific therapies for those specific genes or like those gene mutations. But for the future, we'll have more recognition, so we'll know about more oncogenes. There's already more that I didn't include in this presentation, but as more research occurs, there will be more oncogenes be uh, acknowledged, and then there will be more therapeutic options with more drug discovery. But then we'll also move on to like future kinds of medicine. So I didn't use any like research for this specific part of the presentation, like specifically this line. But for example, like we don't use microRNAs for transcription inhibition as like a therapeutic use, but maybe in the future something will change or we'll develop a new protein that will allow us to use microRNAs as like a better therapeutic as a better therapy. So then we'd be able to inhibit transcription rather than inhibiting like a protein function. And then there's been a lot of money going into AI, which predicts protein folding. So once we are very good at predicting protein folding, we'll be able to design proteins. And then once we can accurately design proteins, we'll be able to do things like transcription inhibition in like a different form, right, rather than microRNAs, or even protein target inhibition using proteins that we developed. So Google's AlphaFold is like in the news a lot right now. It's had a lot of money go into it, but once again, like, we're not able to actually use it for any therapeutic uses at the moment. Right. And here are my references. Thank you.